Hey guys, today we're going to be talking about human genetics and pedigrees. Basically, human heredity involves the same relationships between alleles, such as dominant and recessive interactions, polygenic traits, and sex-linked genes. Inheriting traits is a complex process. Several genes can control one trait, such as eye color, which is controlled by at least 3 genes. However, even though many genes control a person's height, the environment a person is in during their growth and development is a larger factor in height. Recall that genetic disorders are carried by autosomal genes. If someone is a carrier of the autosomal gene, the disorder doesn't show the disease, but it can be passed on. Whereas both males and females can be carriers of autosomal disorders, only females can be carriers of sex-linked disorders. Most disorders are located on the X chromosome, so males don't have a second normal allele to mask it because they are a XY genotype. This means that one copy is enough for the males to have the disorder, and they can't be carriers. Therefore, females can be carriers because they have 2X chromosomes, meaning that they can have a second allele to mask the disorder. If only the mother has the allele and is a carrier, a child has a 50% chance of inheriting that allele. The daughter wouldn't show the phenotype, but the son will.
Since we know that mothers are carriers of some sex-linked disorders and some fathers show the phenotype, pedigree charts can help trace the phenotypes and genotypes in a family to determine whether people carry recessive alleles. When phenotypes are known, genotypes can be determined. On a pedigree chart, boxes represent males and circles represent females. A shaded shape means that the person shows a trait. A white shape means that the person does not show the trait. And a half-shaded shape means that the person is a carrier of the trait. The lines connect the person to their mate and children. In order to understand the pedigree, you also have to use your own logic and reasoning. If approximately the same amount of males and females have the phenotype, then the gene is most likely an autosome. But if the phenotype is more common in males, then the gene is probably an X chromosome. Tracing autosomal genes is simple. Basically, if a person does not show the trait, then they are homozygous recessive, because a homozygous dominant or a heterozygous genotype would show the phenotype. For example, take a widow's peak hairline where the hairline has a downward point. This is a dominant trait. So people who have this hairline either have a homozygous dominant genotype or a heterozygous genotype. If someone does not have a widow's peak, then their genotype is homozygous recessive. If two homozygous recessive people were married, then their kids wouldn't have a widow's peak hairline. However, if two parents are heterozygous for this trait, then it is possible for their child to be homozygous recessive for a widow's peak hairline.
Tracing sex-linked genes is a bit more complicated. You have to think about the inheritance of the sex chromosomes as well as dominant and recessive alleles. An example of a sex-linked trait is red-green color blindness, which is a recessive allele. The genes for color vision are on the X chromosome. In order to trace the sex-linked genes, you have to think about individuals on the pedigree chart. In order for a female to be colorblind, they need to be homozygous recessive. Males who are colorblind must have the recessive allele. Females that are heterozygous for their alleles don't show the phenotype, but are carriers of the trait and can pass it on to offspring. A female carrier and a male with normal color vision have a 50% chance that their son will be colorblind, and a 50% chance that their daughter would be a carrier. Colorblind females and males with normal vision will have daughters who are carriers and sons who are colorblind. Two colorblind parents will always have colorblind children because both of them only have the recessive allele.
Alright, so the human genome, or basically the entire DNA in a human cell, is really large and makes mapping genes difficult. Pedigrees are useful to study genetics in a family, but karyotypes are pictures of all the chromosomes in a cell. If scientists want to study chromosomes, chemicals are used to stain them. A pattern on the band of the chromosome is produced. The locations and sizes of the bands are consistent for each chromosome, but the bands themselves differ greatly. This way, different chromosomes may be identified easily in karyotypes. Besides determining chromosomes, karyotypes can show changes in them as well. Wow, that was a mouthful. Take it away, Ira.
Thanks, Anushe.
For example, a person may have too many chromosomes. In Down syndrome, a person has an extra copy of a part of chromosome 21, or a karyotype can show the loss of a part of a chromosome. Scientists also use karyotypes to determine distances between genes on a chromosome. Chromosomes can be searched to find a particular gene by cutting them apart into smaller pieces. Then these chromosome parts are searched to find the gene. It's a slow and inefficient method, even though many genes and their locations are found along the way. So, just to make sure everything was understood, pedigrees are charts that help trace phenotypes and genotypes in a family. This determines whether the person is a carrier of a recessive allele. A karyotype is a picture of all the chromosomes in a cell. The chromosomes are studied by being stained with chemicals. Both these methods help determine human heredity and gives a person a better understanding of the genes on chromosomes.